First plenary lecture of this symposium. May I now request Dr. Pavan Malhotra to please chair the plenary lecture by Professor Atsushi Yokota. Dr. Pavan Malhotra is the head mammalian biology, Malaria International Center for Genetic Engineering and Biotechnology, New Delhi. He is currently working on functional genomic as well as proteomics of malaria parasites, as well as on the insect cell line from Spodoptera fugiperda. He is a member of the Department of Biotechnology India Task Force Committees and a member of the Guha Research Conference, India. He is a recipient of several awards, including the Krishnamurthy Award in 2009 by the Society of Biological Chemists in India. He has more than 90 publications in peer-reviewed journals and has filed 5 patents. May I request you, sir, to introduce Professor Atsushi Yokota.
It's my pleasure and pleasure for the foundation to welcome Professor Atsushi Yokota, who is a professor of microbiology and Dean University, Hokkaido University, Hokkaido, Japan. And we are very grateful, sir, to have such prominent speaker with us today. Dr. Yokota actually has been awarded by Society of Biotechnology Japan in 2012, and also he has been awarded by the Japan Society for Bioscience and Biotechnology. He has a very prominent speaker, and he has published around 125 original papers in peer-reviewed international journals. He has also reviewed on about 15 book chapters. His main contribution has been to understand various host factors which actually control the gut microbiota composition and also regulate disease progressions. And as you know that some of these, there is a balance between the host factor and the microbiota, various kinds of microbiome in the intestine, and that balance has to be maintained within the gut so that disease progression is checked. So today's talk will be one such host factor, bile acids. And as you know that bile acid is an important factor that regulate various physiological aspects in the body. So I will not take much time and invite Professor Yokota for his prominent talk today among all of us. Welcome.
Well, thank you very much, Dr. Professor Malhotra, for the nice introduction. And it is my pleasure to be here as the first speaker of the plenary lecture of this symposium. First of all, I would like to thank Professor Ganguly, the president of Yakult India Microbiota and Probiotic Science Foundation, for inviting me to this symposium. And thanks are also due to all the staff members involved in this symposium, including Professor Nail and Dr. Hachela. Okay, I hope I can contribute as much as possible to, for this symposium to be successful. My topic today is bile acid as a host factor to control gut microbiota composition and disease development, which we have been conducting this work for the past 2 decades. So my talk is divided into 5 parts. First 2 parts are introductory parts, And the third part is a main body, which we have clarified recently. The bile acid is a host factor that regulates the composition of the rat fecal microbiota. And the chapter 4 is a discussion part, and the last chapter is the recent progress in understanding bile acid as a mediator of disease development, as well as our recent progress in this direction. So let me start. Okay, so recently the idea that gut microbiota has influenced the host health or obesity become popular due to the work done by Professor Gordon's laboratory in Washington University. Here I have listed 3 representative work published from his laboratory, and all are dealing with the imbalanced gut microbiota population, in all cases associated with obesity. In mice or human trial study, indicating the increasing firmicutes, which is gram-positives, and the decreasing bacteroidetes, which are gram-negatives. And I will explain the third one, this one, as the representative work from this category in the next slides. So in this paper, adult mice were fed the standard diet or high-fat, high-sugar Western diet for 8 weeks, and Western diet induced obesity. When we look at the gut microbiota population in the standard diet, here, balanced population between the firmicutes and bacteroidetes was observed at the proportion of 75% firmicutes and 20% bacteroidetes. In contrast, the Western diet altered fecal microbiota community in a way that firmicutes was enriched at the expense of bacteroidetes. So after 8 weeks administration of the high-fat diet, the microbiota was predominated by firmicutes at the proportion of more than 90%. So let me move on to gut microbiota and disease development. And recent study have shown that the gut microbes are associated with not only with obesity, but also many metabolic diseases, such as metabolic syndrome, including type 2 diabetes, IBD, inflammatory bowel disease, IBD, and Crohn's disease, and colorectal cancer. And so we can probably categorize these studies into 2 different directions. The first category probably is clarification of gut microbiota alterations induced by high-fat diets or disease development. And the second category may be the clarification of how the altered gut microbiota develops a metabolic syndrome, IBD, colorectal cancer, etc. So we now know that high-fat diet or Western diet induce imbalanced gut microbiota population or dysbiosis, as well as metabolic disease induction. And people are now trying to elucidate the causal relationship between imbalanced gut microbiota and metabolic disease development. But I think the program is underway, and in my opinion, what has been completely lacking in these studies seems to be the mechanisms that reshape gut microbiota in response to high-fat diets. In this context, we have hypothesized that bile acids might be the host factor that reshape gut microbiota composition on high-fat diets. So in the next chapter, I will introduce bile acid in special reference to its antimicrobial activity. So bile acids, antimicrobial activity. So bile acids are sterol compounds synthesized from cholesterol in the liver, excreted into duodenum as a main component of bile. And bile acids help emulsification and absorption of dietary lipid by acting as detergents in small intestine. And bile acids are excreted in larger amounts on administration of high-FODS diets. And besides these digestive function, bile acids also have strong antimicrobial activity due to their detergent effects on bacterial cell membrane, and thus seem to exert strong selective pressure on the gut microbiota. And bile acids structure, metabolism, antimicrobial activity. So here you can see the structure of the typical bile acid in humans. And bile acids are synthesized in the liver as a conjugated form, bile acid, free bile acid, and the taurine or glycine conjugate. And the bile acid underwent biotransformation during the transit down to large intestine, which includes the first the deconjugation reaction with bile salt hydrolase activity, which splits the bile acid into free bile and taurine or glycine. And after that, free bile underwent 7-alpha dehydroxylation reaction, which eliminates 7-alpha hydroxyl group to yield deoxycholic acid from taurochloric acid. This is a representative bile acid in human. And then by way of cholic acid. Since these reactions are quite efficient, so that the deoxycholic acid predominate in large intestine or fecal sample. And as these reactions increase the hydrophobicity of the bile acid molecules, so the deoxycholic acid has a very, very strong affinity to biological membrane, exhibiting a very strong antimicrobial activity, which is 10 times higher than cholic acid. So this is more hydrophobic than this one, so it has a very strong antimicrobial activity. We have previously determined the MIC, minimal inhibitory concentration for these bile acids, and found that the cholic acid, for example, has a 5 millimolar to many intestinal bacteria. In contrast, the deoxycholic acid has MIC value of 0.5 millimolar, which actually means 10 times stronger bactericidal activity than cholic acid. So please remember this value for the rest of my talk. Anyway, these free bile acids have strong antimicrobial activity due to the membrane damaging effects. Okay, so let me move on to the main part. Part 3, bile acid is a host factor that regulates the composition of rat fecal microbiota. Okay, in order to clarify the effects of bile acid on the gut microbiota composition in vivo, we have conducted a simple experiment where rats were fed controlled diet and bile acid supplemented diet at two different concentration of cholic acid. One is physiologic concentration, MCA, and this is non-physiologic, very high concentration of bile supplemented diet, HCA, for a short period, 10 days. And after that, we have collected these biological samples for analysis. And here I would like to focus on the bile acid analysis using fecal samples and also microbiota analysis using fecal contents. So what you can see here is bile acid metabolism in fecal sample after 10 days of bile acid or choric acid feeding. So blue color represent control and red MCA, light green HCA sample. So as you can see here, choric acid increased dramatically in the fecal sample. This is quite natural because rats were fed choric acid. But at the same time, we found that the deoxychoric acid increased significantly. This means that the choric acid has been intensively converted into deoxychoric acid by gut microbes. And we have calculated the deoxychoric acid concentration in cecal contents. As shown in this table, the cecal contents of the choric acid fed group contained deoxychoric acid at the concentration more than 1 millimolar, which is already reached very, very toxic level to kill the intestinal microbes. Please recall the value 0.5 millimolar for deoxychoric acid. So we can instantly know that these deoxychoric acid concentrations or the presence of the deoxychoric acid exert a strong selective pressure on gut microbial community. So this is how the cecal microbial communities look like after 10 days of cholic acid administration. So the control group showed a variety of morphology from cocci, short rods, and long rods. However, in choric acid fed group showed a much less variety, largely displayed by cocci or short rods. These results suggested that the bacterial population in the choric acid fed groups are altered. So as expected, the sequencing 16S rRNA gene clone libraries revealed striking alterations in the bacterial population in the choric acid fed groups. Our results show that, that the bacterial community in the control group showed a quite balanced population with Firmicutes 54% and Bacteroidetes 30% and the rest minority groups. These values are quite similar to what has been observed in human and mouse experiments. In contrast, choric acid fed group showed a dramatic expansion of Firmicutes, which reached more or less 95%. So the Firmicutes increased and the Bacteroidetes almost disappeared. So this is a class level alterations. The increase of the Firmicutes was due to the expansion of Clostridia and Erysiperotorchi class of bacteria, while Bacteroidetes disappeared. And this is the genus level alterations. The Clostridia expansion was due to increase of genus Blautia, and the Erysiperotorchi increase was due to genus Arobacillum. So Blautia, Arobacillum increased, and Bacteroides disappeared. So these are the alterations, and then we have analyzed the diversity of the microbial community by OTU, operational taxonomy unit analysis, using a 97% nucleotide identity as a cutoff. So we identified 117 OTU, and it was revealed that the specific bacteria at the genus or species level were highly concentrated in cholic acid fed groups like this. And accordingly, the diversity assessed as Shannon index was decreased in the choric acid fed group like this as compared to the control. So these results clearly indicated that bile acids are host factors that determine gut microbiota composition in vivo. So in the next chapter, I would like to discuss these results. And here we raise a question, is bile acid the determinant of the gut microbiota on a high-fat diet? So I would like to here compare our result with previously obtained gut microbiota alteration obtained on a high-fat diet administration. So our result is listed on the top row, where rats were fed choric acid supplemented diet, and we found increased Firmicutes to Bacteroidetes ratio, and also the decrease of the biodiversity and expansion of Clostridia class, Erysiperotorici class, and genus Albaculum, Clostridium inoculum expansion. So these results were quite similar to what have been obtained on a high-fat diet feeding to mouse or hamster experiment, where we can see again the increased Firmicutes to Bacteroidetes ratio, and also the decrease of the biodiversity and expansion of the very similar phytotype member. So probably these results strongly suggest that the bile acid serves as the important determinant reshaping the gut microbiota on a high-fat diet. And we propose this idea as bile acid hypothesis. Although we are still on the way of validating this hypothesis, there are several papers recently published which can be interpreted readily by applying this hypothesis. So I will introduce two papers in this direction. First paper is the result which compare the fecal microbiota of children in rural Africa and urban Italy. So African children are administrating traditional rural African diet, which is quite similar to vegetarian diet, as characterized by low fat and animal protein, rich in starch, fiber, and plant polysaccharides. So under these conditions, it was found that the gut microbiota was dominated by Bacteroidetes, in which Prevotella predominates. And Prevotella has been found abundant in rumen of cow and sheep for health-degrading plant fibers. So probably these bacteria are working to degrade plant polysaccharide or fibers in the diet. And biodiversity found to be quite high. In contrast, children in Italian urban Italy are administrating, of course, the typical Western diet as characterized by rich in fat, animal protein, sucrose, starch, and low in fibers. Under these conditions, Firmicutes expanded and at the expense of Bacteroidetes, and in Bacteroidetes, Bacteroides replaced the Prevotella. And also the biodiversity has decreased. Here I would like to apply bile acid hypothesis to these observation for much easy understanding. So from the composition of the diet, we can assume that the lower intestinal excretion of bile acid in African children as compared to Italian children. And it has been reported that bile acid resistance is higher in Bacteroides than Prevotella. These facts, together with the availability of the carbon sources in the intestine, works as selective pressures to switch microbial community from Purebatora-rich one to Bacteroides-rich one. So this is my interpretation. Another example is the enterotypes proposed by researchers in the MetaHit consortium in Europe. They found that intensive sequencing analysis of human gut microbiota can identify 3 robust clusters refers to as enterotype, including Bacteroides enterotype and the Prevotella enterotype and Ruminococcus enterotype. And following this work, Wu et al. Correlated the occurrence of these enterotypes with diet. And they found that the Bacteroides enterotype is associated with Western diets and the Prevotella enterotype with carbohydrate-based diets. So, again, I would like to apply bile acid hypothesis that the bile acid excretion in Western diet is higher in intestine than carbohydrate-based diets. And again, the bile acid resistance of Bacteroidetes is higher than Prevotella. So selective pressure of bile acid might contribute to establish these enterotypes. Okay, so here comes the last part, recent progress in understanding bile acid as a mediator of disease development. In this section, I would like to introduce 2 recent papers describing these aspects, as well as our recent progress in this direction. So, first example describes the taurochloric acid as a mediator that provokes colitis in mouse intestine. So, in this experiment, mice were fed 3 kinds of diet. The low-fat diet, this is a control diet, and the high-fat diets, polyunsaturated safflower oil-derived fat supplemented PUFA diet, or saturated milk fat-derived fat diet, MF diet, for 3 weeks. And as a result of this feeding, a bloom of sulfate-reducing bacterium, Birophila batsotia, was observed in milk fat diet, as shown in this yellow sector in the pie chart. And interestingly enough, milk fat, but not low fat or PUFA fat, increased the incidence of colitis in genetically susceptible IL-10 knockout mice. So here, the milk fat diet induced the colitis, but not in low-fat and PUFA diet. And the authors found that the milk fat increased the taurochloric acid proportion in the gallbladder bile because of its hydrophobicity containing C16 saturated palmitic acid. And also, the authors found that the taurochloric acid promotes the growth of Birophila batsotia as this bacterium uses taurochloric acid as the source of taurine, which is used as a terminal electron acceptor during the anaerobic respiration and yielding hydrogen sulfide. And the authors speculated that the formation of hydrogen sulfide may increase the permeability, mucosal permeability, and perturb immune responses, and leading to the colitis development. And these results indicated that taurochloric acid as the mediator for colitis development. So another example is a liver cancer development mediated by increased serum DCA concentration during high-fat diet intake. In this experiment, wild-type mice were fed normal diet or high-fat diet for 30 weeks, very long time period. in the course, DMBA, which is a chemical carcinogen, was applied once at the very beginning of the feeding. And as a result of this, mice fed high-fat diet became obese and developed liver cancer, as shown here, which was not observed in the mice fed normal diet. And on analysis of the gut microbial community, as usual, the increase of the firmicutes and decrease of the Bacteroidetes were observed. And especially the gram-positive bacteria belonging to Clostridium cluster 11 and 14A related to deoxycholic acid formation were bloomed. And analysis of the serum bile acid composition revealed increased deoxycholic acid proportion, actually. As deoxycholic acid is already known as the carcinogen, which stems from the DNA damaging effect along with reactive oxygen species production. So DCA is known as not only as colon carcinogenesis, but also the liver carcinogenesis. So the authors tested the effect of DCA feeding to check if DCA is responsible for liver carcinogenesis. In the experiment, mice were fed high-fat diet with deoxycholic acid for 17 weeks together with antibiotic treatment to eliminate the effect of gut microbes. So DCA feeding, as a result, developed liver cancer here, suggesting that DCA alone was sufficient for the liver cancer development. So these results clearly indicated that high-fat diet altered gut microbiota composition to increase serum DCA concentration, which in turn triggered liver cancer development. So besides these findings, the bile acid has recently been clarified as signaling molecule to the host bile acid receptor FXR, for example, that works as a nuclear receptor and regulating all kinds of these host bile acid and the cholesterol and lipid glucose metabolism. So therefore, bile acid can be the target molecule to be looked at not only as the mediator of gut microbe composition or regulator of the gut microbiota composition, but also the regulator of the disease development, which connects all three domains. So in the last slide, I will briefly introduce the recent progress in my laboratory in this direction, which has not been published yet. Ongoing work. What we have done was, again, very simple rat experiments. Rats were fed choric acid supplemented diet again, but this time 13 weeks, not 10 days. And we have observed increased deoxycholic acid concentration in cecum by bile acid converting bacterial activity, and also, again, the alteration of the gut microbiota composition, increasing firmicutes and decreasing Bacteroidetes. And interestingly enough, we have detected the fatty liver development in choric acid fed group, together with increased liver damage markers and mucosal permeability. So these results clearly indicated that the bile acid directly induced the malfunctioning of liver lipid metabolism and liver damage, and probably underlying the metabolic disease development during high-fat diet administration. And also this animal model has advantages in which we can simplify the experimental system by focusing on the direct effects of bile acids on metabolic disease development while eliminating factors derived from high-fat diet administration. Okay, before concluding my talk, I would like to thank all these people listed in this slide for collaboration in microbiota analysis and animal experiments and bile acid analysis, and also funding from Yakult Bioscience Foundation. So this brings me to the end of my talk, and thank you for your attention.
Professor Yokota, for a wonderful talk. We have time for a few questions, if anybody has. Professor Ganguly.
Dr. Yokota, the very nice paper. In India, there is a problem which is known as NAFLD, which you mentioned, non-alcoholic fatty liver disease, and NASH. And there are some, and this is one of the reasons for, apart from hepatitis viruses, for liver cancer. And there are 2 papers from Indian universities showing that the probiotic reduces NAFLD. These were human studies. So it's very interesting that you have conducted these in the mice. And it links both the studies and the science of this into the genesis of liver cancer. Thank you for this presentation.
Thank you for your important comments.
Next question.
I am Dr. Prajapati from Anand. I have 2 small questions. One thing which you referred to in presentation, humanized mice. What do you mean by that? That is number 1. And second thing, that can you explain some relation between bile acid and the obesity?
So I cannot hear the first one very clearly.
Humanized mice. In your treatments, you had models. One model was humanized. Whether you altered the microphora or what was done?
Well, I don't remember very clearly about that work, but the virus metabolism in humanized mice might be a bit different from rodents' metabolism. But I haven't remembered very clearly that one. And the second one is bile acid and obesity. Well, it's very difficult, and normally the Gordon's group initially insisted that the organic acid production in the intestine accounts for the extra energy gaining, and it may be that the bile acid may reshape the gut microbial community in a way that the organic acid production might be increased by Clostridia-rich microbial community. But it's very difficult to, I think, validate, because the organic acid has a steady state concentration, consumption, and the production. So it's very difficult, I think.
Question from
Sir, Dr. Dinakaran from Chennai. What is the interplay between the bile acid, probiotics, and gallstones?
Gallstone.
Yeah.
I think I'm not the physician, so I don't know very well about the gallstone, but it has been said that the gallstone tend to be formed in people rich in deoxycholic acid contained gallbladder bile. So since the deoxycholic acid is the product of the bacterial biotransformation of the original human bile, so the bile is underwent enterohepatic circulation, so the deoxycholic acid may accumulate repeated biotransformation from choric acid to deoxycholic acid. So it may be that the gut microbes are quite enriched in DCA forming bacteria in gallstone forming patients. This is my opinion.
Hello.
Two last questions.
Yeah, thank you so
I'm
much.
Dr. Mehra from AIMS. My question is related to the Shannock's index that you talked about. And you said it falls from 5.8 in the normals, in the control group, to somewhere like 2.7 or so. Now, Indians are normally primarily vegetarians, you know, their food is low-calorie food. Now, do you think that the Shannock's index, which I think is a very good marker for the effect of bile duct, as you talked about, the bile acid, as you talked about, so Shannock's index will vary from one population to the other. India, with a very diverse kind of foods that we eat, primarily being vegetarians over here.
I cannot get the point that do you...
I wanted to know, that if you want to use this as an index to understand the effect of the bile acid, that will vary from one population to the other, one ethnic group to the other. Because of the type of food habits that they have.
I cannot get the point, but this is just to want to show how the diversity is decreased in our system, and
You think the Shannock's index is a wonderful index for understanding the relationship between the two species?
species variation.
Firmicutes and the bactericides?
Well, this is just the index for how a difference between each sample. So anyhow, here OTU analysis indicated that the only first 3-4 species occupy more than 70 or 80% of
so
the...
that's what I meant. I was translating your experimental data into the actual practice in terms of the food habits.
Mm-hmm. Yeah, we have to assess by PCOR or other way as well.
Yeah, I just wanted to know one thing. I'm Dr. Mohania from Sir Gangaram Hospital. As you said, the secondary bile acids like the deoxycholic acid is responsible for the liver cancer development. But do you have an idea, apart from the liver cancer, is it also involved in the development of other sorts of the organ tumor development like colorectal cancer?
Can you say it a little bit slowly?
Yeah, my question is that, as you said, the secondary bile acid like deoxycholic acid, which is responsible for the development of the liver cancer. I know as far as regarding the colorectal cancer, this is also involved in this. Do you have an idea, apart from the colorectal cancer and the liver cancer, this secondary bile acid like the DCE is responsible for the development of other sorts of tumor in our body system?
Other cancer?
Yes,
Other cancer.
yeah.
Other cancer?
Other cancer.
I don't know.
Other two.
But yeah, I'm sorry, I don't know.
Okay, I think we have passed our time now. We should thank Professor Yokota again for a wonderful talk. I think
So thank you very much.
I will still say the real crux will be that if we can correlate, as Professor Ganguly said, with the human data and mouse data together, that will give us the real fruit that what we are telling may give us the right diet or right supplement to control some of these type of cancer. Thanks, Professor Yokota.
Thank you very much.